Keep everybody. Let me keep everybody informed about what's going on. Today's Cancer Day. Yesterday was Virus Day. Viruses lead, as you saw from Steve Goff, viruses lead naturally into. In fact, it was viruses that really broke open the cancer problem, and they lead naturally into talking about. You now know enough about viruses so we can talk about them facilely and talk about how it naturally leads into a deeper understanding of cancer biology at the molecular level. We have two talks on cancer, and then we had asked Mike Rorschbach, who he has many wonderful attributes. He's a really good speaker. He's worked all his life on circadian rhythms, which are how biological time is kept in cells, which is a really interesting topic, and oddly enough, a little bit related to cancer in a number of ways. Time is is an important variable in cancer. However, it's raining in Boston, and all it has to do is drizzle in Newark, and flights are delayed. So his flight was delayed, and it keeps getting delayed. So certainly what we're going to do is have Raoul and I talk this morning, and then we'll go right in. Instead of the third talk at 1 o'clock or 2 o'clock, Raoul and I will hold our discussion section on cancer, and we'll sort of wait it out and see, when he lands. And we're just trying to find out the absolutely latest time he's willing to lecture here. So far, it's about 12 o'clock midnight tonight. So, so prepare for a long night, everybody. Any way you want. Yeah. Okay. So it is a good habit, I think. It's at least my habit, anyhow. To start with a little bit of history, we started when I introduced the course. I started with Darwin in 1859. I won't go back that far, though some of the experiments are in the early 19th century that we'll want to talk about. But I'll take you back to the year in the 1960s, right? Because in the 1960s, you would have gotten an argument by different scientists about what the causes of cancer were. And they were so different from each other that it didn't seem that there was going to be a unified theory of understanding cancer at all from the perspectives of the 60s. So I'm going to take you back to 1960s and what people were arguing about as the causes of cancer. And I'll take you through a little history of it. And then we'll build from that kind of thing. There's some very interesting things that come out of this discussion that allow us to build. So really the first interesting breakthrough was a virus you heard about yesterday, which was Rous sarcoma virus. It was discovered by a guy named Peyton Rous. Peyton Rous was a pathologist, educated at Johns Hopkins, took a job in 1906 at Rockefeller Medical Research Institute in New York. And his advisor at Hopkins said to him one thing. He said, when you go up to Rockefeller, actually this tells you a little bit about the culture of the days. You're going to meet with Simon Flexner. And Simon Flexner was the head of Rockefeller Research Medical Institute. And he's going to tell you what to work on, right? Now that's different. Directors of institutes don't tell anybody what to work on anymore. And if he says work on cancer, you don't do it because that's the one subject that is so befuddled and so ridiculous nobody could figure out what the heck's going on. However, so get an idea or two before you go up and fight for your ideas. So he went up and Simon Flexner told him he had to work on cancer. And and he was a chicken, so he worked on cancer. And then the chicken really came to roost for him. So there was a farmer in New Jersey, actually, this is the hero of the story. The farmer in New Jersey who had a chicken, and the chicken had a tumor. And he brought the chicken with the tumor over to Rous in New York. And he put the chicken on the lab bench and who ran around and everything. And Rous was asking him questions about it. And then he heard the farmer say, all my chickens have this tumor. It's not only this chicken, all the chickens have this tumor. Now, it was actually a breast tumor of the chicken, but it was the muscle of the breast in the chicken, and so it's called the sarcoma, right? So carcinomas are epithelial tumors, sarcomas are mesenchymal tumors. That's the germ layer that makes muscle and bone and so forth. So there's a whole series of tumors called sarcomas, and they're all from the inner mesenchymal. The blood is mesenchymal, for example, right? And Rous was taken aback by the idea that this chicken could have an infectious disease that causes cancer. Now, actually, oddly enough, Goff told you about an infectious disease that causes cancer, which we know about. But he was taken aback. And in those days, viruses were all the rage because, as Goff told you, or actually I think Tom Schenk told you, in 1889, a Russian scientist discovered the first virus, tobacco mosaic virus. And the way you knew it was a virus is that you poured it through a filter that stopped all the bacteria, which are about 1 micron big, and so you're looking at things smaller than a micron. It came through, and then you took the material that came through the filtrate, and you inoculated it into an animal or a tobacco plant, and it caused the tumor, right? And tumor, or it caused a virus infection. So Rous decided to do the experiment, actually. He said, if this is infectious, maybe there's a cancer virus here. And he filtered it, took the tumor, he broke it open, he killed off all the cells, he filtered it, so he has no bacteria in it at all. He inoculated the chicken, and sure enough, chicken got a tumor right at the site of the injection. Then he could pass it, and he could find it, and he could filter it again, and he used Koch's postulates to show that this virus caused cancer, right? And in 1911, he published a paper, and the virus came to be called Rous sarcoma virus, or RSV, right? Now, historically, that becomes a very important virus in the chain of things to come, and I'll come back to Rous sarcoma virus. But that demonstrated something that then everybody started looking for. Do viruses cause cancer? And it got solidified. There were a number of viruses that could cause cancer. So we thought at the time, that's pretty clear, viruses cause cancer. However, in Japan and in elsewhere, in Wisconsin, for example, about the same time, a group of people started using chemicals. They painted chemicals on rats or mice, the skin of rats or mice. Historically, if you really want to go back to the 16th century or so, about 16th, 17th century, there was an Englishman named Sir Percival Pott. And Sir Percival Pott figured out that chimney sweeps, who were little boys, chimney sweeps were little boys, they lowered down the chimney, right? And they swept out the chimney, and they got testicular cancer, right? And so he actually hypothesized that maybe there was something in the material in the chimney that was actually causing the testicular cancer as they lowered the chimney sweeps down. In the medieval times, it was found, that was reported by a bunch of monks, actually, that the women who were nuns, and therefore never married, never had children, women who were nuns got breast cancer at a higher rate than women who were having children, right? And all of these kinds of epidemiological observations and things were coming to the conclusion that maybe chemicals were important for cancer. And this Japanese group, Yamagawa in particular, found a number of chemicals that if you paint on animals, the skin of animals, they would form tumors on the skin at the site of, right? And these were called carcinogens, and it was pretty clear chemicals could cause tumors, right? And then NIH got formed. The National Institutes of Health got formed in 1945 or so, and they, one of the institutes of health was the Cancer Institute, and they hired epidemiologists who started looking for all sorts of associations of cancer and causes of cancer. male, female differences which are found. What job you had was important, could be an important variable. And they did an experiment that I'll talk about a little bit more detail, demonstrating rather clearly that one of the really remarkable variables was age, right? That with time you'll see there's 3-4 log increase in the frequency, in the incidence of cancer in all animals. Forget about just humans. All animals as a function of age. Animals in age is a very curious thing if you think about it, because all animals live different lifespans. So we have a biological age. Since all animals live different lifespans and they're different species, it's pretty clear that aging is a program and that death is a program. It's programmed. It's actually programmed genetically and epigenetically. And we'll have a whole day on aging and what it is and all the different ways people think about aging, but I'll really bore in for something like cancer. But this is quite a variable. There's 3-4 log increase in the incidence of cancer as a function of age.
Maybe just a sort of question. So since when did people start to distinguish between cancer and just a normal tumor?
The tumor is a cancer. There are many kinds of tumors. There are tumors that are classified by tissue. I told you there's carcinomas, right? I told you there's sarcomas. And there are benign tumors. Yes, right? And if I say benign tumors, that means it's a tumor. It grows. A wart is a benign tumor. So you will have, any of you who had warts, it's a hyperplasia, right? Hyperplasia means a growth, abnormal growth of the skin. It's a well-differentiated wart, right? It has a lot of keratin in it. It's well-differentiated, and it's benign. It's not going to kill you, right? If I say carcinoma, that means it's a malignant tumor. If I say sarcoma, it's a malignant tumor. If I say benign tumor, it's a benign tumor, okay?
And since when did people start to distinguish between?
Oh, I have no idea.
Because all these experiments,
Yeah.
it's not enough that you just observe some physical oddness there.
Stay cool, all right? Just, it's enough for now, okay? It'll become obvious to you that what the causes are and why there are benign tumors and why they're not.
Sure that benign and malignant tumors were distinguished by data.
Oh, that may... look, the Greeks understood what tumors were. They even operated on, took them out, right? But they didn't understand the difference between benign tumors and malignant tumors, right? You wouldn't begin to understand what a benign tumor and malignant tumor are caused by until I get there, okay? So that's why I want you to just not worry about it for a while. Because the same genes cause that. Exactly the same genes. And the same gene can cause a malignant tumor and yet also cause a benign tumor of another tissue, right? So it's more complicated than say that you have to define it. You have to understand it, actually, rather than define it. Okay, so the first problem we faced in the 60s, and that's why I bring this up, is these are 3 very disparate kinds of biological properties, chemicals, viruses, and aging, right? And that really messed up the field, to put it neatly. For those of you looking for research problems, just put yourself back into the 1960s and know these 3 facts and ask for a unified idea about what causes cancer. And if we just take one moment to think about what's common about these 3 things, you might actually be puzzled, right? So unfortunately, the fields divided up and never spoke to each other. So there were chemical carcinogenesis guys and there were ladies and guys, actually most of the guys in those days, but chemical carcinogens, ladies and guys, and then there were virologists, ladies and guys. And then there were aging specialists, ladies and guys. And they had their own meetings, they had their own journals, and they never talked to each other. Right? So the idea of unifying these things never occurred to people. I'm going to go back to this theme of making observations and then asking you to unify what seems like disparate things and then build to it. But give it some thought as we go through it. Okay. So the aging paradigm, though, could be quantified. That is to say, we know how old you are biologically. Now we're going to have to define aging. That's not a bad thing to define. There's chronological age, right? You know your birthday, usually. If you know your birthday, you can count, and you know the number of years you lived, and that would be chronological age. I'm going to be talking a little bit about biological age, which may be a percentage of lifespan, right? Or we're all aging at different biological slopes of aging, right? So age is a little bit more complicated than just saying chronological age, but for the time being, we'll stick with time as chronological age. So what the equation that describes the aging phenomenon is the incidence as a function of time is equal to time. There's equal to a constant, which allows you to write the equation. It's equal to time times r to the minus 1, right? And no one quite knew what r was, but with time, you'll find out that r is the mutations, is the number of mutations that you'll find in a tumor that are causing the cancer, okay? R minus 1. Yes, not to the minus 1, r minus 1. All right. Now, k turns out to be a really interesting constant, all right, because k is different for different animals, right? So, a dog may live to 10 to 12 years old, and a human may live to 70 or 80 years old, right? And so there's about a 7-fold difference in k in dogs and humans, right? But humans have 4 log increase in the incidence of cancer as a function of the last quarter of their lifespan, and dogs have 4 logs incidence in the of cancer increase in the incidence of cancer as a function of their lifespan, and the only difference is k, right? Yeah.
Does a lot of this incidence across species collapse?
No, it doesn't. So I'll show you dogs, but it's true for mice, rats, dogs, humans. It's true for any animal we know of. The incidence of cancer rises as given by this binomial equation. It rises as a function of time. And it's usually the last quarter of your lifespan in which this happens. Now, there are exceptions to that. If you inherit a mutation, the curve will move to the younger ages, right? And you'll see that, yeah. Okay, yep.
Is this a rate equation, or is this just like the
incidence? It will be a rate equation, yeah. All right, so let's just look at it. This is data, real data, right? This is humans. This is the kind of thing that the epidemiologists looked at. Let me just say that there's another thing. This is the exact same curves you will see if you plotted lifespan, right? And it's not because cancer is the predominant reason you're dying. Your predominant reason you might die if you're a human is infectious diseases. That would be the most common. Cardiovascular would be second, but cancer would be third. If I just displace this curve by about 4 or 5 years for men and women, it would be the same. Women would live longer than men, and both curves would displace by 4 or 5 years. So actually, the incidence of lifespan incidence, or you're in a cohort when you do lifespan incidence. You're put usually in a 5-year cohort, and then your cohort is carried through for the time period. So there's a window that is used in that. But the curves are the same. But this happens to be the percent. In this case, it's percent cancer. It's not rate. So you're increasing the percentage of cancers pretty dramatically, and women are slower than men. And that's true for lifespan as well. Right? And lifespan is not, in this case, dependent upon this incidence of cancer. So that's kind of curious. But that's how we define, of course, one of the last quarters of cancer. So that's sort of the kind of curve that you would see. It's sexually dimorphic, and I could tell you right away that we don't understand really the reason why women are later than men. Because women are usually more advanced than men in many ways. So, okay, just to show you, I've just plotted here the data for. It's racially independent, by the way. So it's not heavily environment or heavily genetic variation. So this is men, USA, men, Japan, and male dogs, right? If you're going to do men, you have to do male dogs because female dogs will get cancer after male dogs get cancer. Female mice will get cancer after mice will get cancer, after male mice get cancer. And age holds the same too. Female mice will live longer, female dogs will live longer. Dogs are a little bit odd because big dogs and small dogs have different lifespans. That's not usually the case. Big people and small people don't usually have. Right? But in dogs, that's the case. So, and here the K is just normalized for dogs, right? So the K is 1/7 for the dogs. And that's just true for every animal we could look at and investigate and things, right? So there's something funny and universal about this aging curve and something very fundamental here, but nobody had a deep understanding of what this meant, right? Or how to unify things. Yes, Victor? It's pretty straight, actually. If I plotted it on a log scale, it would be straight.
What is R?
R is mutation, number of mutations. Oh, 4. 4 or 5. So R is different for different kinds of cancers, first of all. And that's not surprising. So by different kinds, I mean different tissues. R is different for benign versus malignant cancers. That's something you'll now start to see, that you can define benign and malignant by how many mutations you might have in this series. And I'll give you a very good example of that in colon cancer.
So if we plotted for different kinds of cancer, we'll see well-separated straight lines with different values of R close to integers, or how do we do that?
That's a good question.
So it's a straight line in a log-log scale.
Yeah.
Yes, on a log-log scale. Yeah, that's not surprising. Log-log scales are, yeah. So as an approximation, yes. I don't know that it's exactly that. There may be exceptions, but the real answer is I don't remember seeing people actually answering the question you're asking. But I have seen this as a function of a tumor and as a function of all tumors, and the curves aren't very different. They're straight lines in both cases. But if you plot log-log, yeah. And you're talking about almost 4 logs in the change. It's really quite a dramatic difference as a function of age. Something about age that really is very annoying in this case. Okay, so this is a sort of curious aspect of this, that this is relative time, right? This is biological time. It's part of the program. It's almost part of the program, right, that these animals have. And we'll address that kind of thing. You could, yeah, come and sit up here. All right, so this is a good example of inherited. So one of the things that became apparent, I'm going to add a 4th variable to the list. One of the things that became apparent as people studied epidemiology was that in animals as well as in humans, cancer could run in families. And it could behave in a dominant or a recessive fashion. And this is an example of a recessive gene that behaves like a dominant gene. This is a violation of Mendel in many ways. A recessive gene that behaves as a dominant gene in families. So circles are females, squares are males. If you're shaded in, you develop cancer over your lifetime. So you could see 3 generations of cancer. You could actually use this in Mendelian kinds of segregation to actually map the genes. That's pretty rare in humans because you don't usually have very large populations to do the mapping. But in animals, it wasn't rare at all. So it was pretty clear that the genes could cause cancer. Genetic, or presumably genetic alterations. All this was before DNA sequencing. So it wasn't possible to really say that it was a mutation in the DNA, but that's what it's going to turn out to be, and that's why R turns out to be a mutation. R-1 is mutations, right? What would happen in this family, if we plotted this family as a function of age and cancers, is that the curve would move to the left. So it's not surprising that if there was a family of dogs that inherited the mutation because they were inbred dogs, their curve would move here, right? And that would be R minus 1 with one less mutation, right? And that really suggested, if we were thinking about it that way, that a certain number of mutations were required to cause the tumor, but if you inherited one, you would get it at a younger age, right? So that what aging was starting to look like was the time it took to accumulate mutations, right? And the commonality you'll see between all these things are mutations, that the chemicals are mutagens. They're not only carcinogens, they are mutagens. And that some of the viruses that you'll hear about, like the retroviruses, are genes which are carrying mutations. All right? So the unity of this idea really broke in the 1970s. And it broke for two reasons. One, there were a couple of smart people that did the experiment that I'm going to tell you about. That's Bishop and Varmus. They were at UC San Francisco. And the second is that really they couldn't have done the experiment without the kind of technology that just broke open in the 1970s. The ability to clone genes, the ability to sequence genes, the ability to isolate pieces of DNA, which we've taken for granted right up until now. But in the 1970s were new technologies that were applied very rapidly, right? And they were applied, I'm going to now just tell you a little bit about their experiments because it's interesting to hear how it came about that we began to understand this group of genes that are important for mutations that give rise to cancer, right? Rous sarcoma virus plays a big role. So Bishop and Varmus worked on Rous sarcoma virus. And it causes sarcoma, and it is a retrovirus, so it has a genome that's RNA, right? And in those days, you could run out the RNA and you could size the genome, and so we could know how many kb it was, right? So Rous sarcoma virus, right, was about 10 kb. Right? And what Goff told you, and what we kind of knew at the time it was building, was that it had a GAG gene, right? It had a POL gene, and it had an M gene, right? And then there was a second virus of chickens that looked exactly like that in every way. So the RNA looked very similar, the reverse transcripts looked similar, everything looked similar. The ability to clone it and everything looked similar, but it didn't cause a sarcoma. It caused the leukemia. So these were called avian leukemia viruses, right? And it had an RNA that was about 8 kb long, about 8,000 base pairs long, and it had a GAG gene, and it had a POL gene, and it had an M gene. Right. And as the Bishop and Varmus began to sort of look at these two different viruses, one of which caused a sarcoma, a malignant tumor of the breast tissue, and one of which caused a leukemia, a malignant tumor of blood cells, right? Very different tissue specificities. There was 2 kb extra of DNA, about 1.6 to 1.8 kb extra of RNA in this virus, right? And so they decided they would clone that 1.5 kb and ask the question, is it possible that that was the reason why one made a solid tumor and the other one made a liquid tumor, if you will? So what you do is you do something called subtraction cloning. Right? So I make single-stranded DNAs of each. I hybridize those single-stranded DNAs together. I get double-strand, and this gives me double-stranded and single-stranded DNA. Right? I get myself a little nuclease. That's an enzyme that degrades DNA, but it only degrades double-stranded DNA, and it degrades the double-stranded DNA, and I'm left only in the test tube with single-stranded. I make this second strand. I put it in a plasmid, I put it in E. coli, and I've cloned this extra piece of DNA. So, did I say that too fast? Does everybody follow? All right, I'm just taking the piece of DNA and I'm isolating it, purifying it, growing it in a foreign organism, E. coli, right? And I grow up a lot of it, so I have a lot of a little piece of DNA. Okay? Yeah.
So what is the part of the isolation, the one that you want to grow?
I'm isolating this single strand, right?
Part of the M?
No. I'm looking for, yeah. So when I made a double strand, right, and I used the nuclease, here's what happened. Right? So what I'm cloning is the piece that's in the Rous sarcoma virus that's not in the avian virus. Right? It's a subtraction method, right? Okay, so yeah. Yes, yes. So this is covalently bound. It's a single RNA molecule, just like all retroviruses. I'm just manipulating it now with enzymes and degrading it and so forth and so on. So I've gotten rid of all of the GAG, POL, and M genes because they're in both. And you know that GAG, POL, and M is needed for them to replicate, right? Okay. Right. Now, this DNA was then, for the first time, sequenced. By 1976, we could sequence DNA, right? So it was sequenced, right? And there was a big surprise when it was sequenced. First of all, we didn't really have databases in those days. There were very few things sequenced. So you got a sequence and you looked at it and you could make a protein out of it, right? And you didn't know much more than that, right? So they said, well, where does this sequence come from? It obviously is not necessary for the virus because the avian virus replicates perfectly well without it, right? So where did it come from? Maybe it came from the chicken, right? So they hybridized it to chicken, and it did. So this extra DNA came from the chicken, right? Now, Goff told you how it could come from the chicken, right? If you had an avian virus, avian leukemia virus, and it integrated into the chromosome, it could have integrated next to this chicken DNA, and then when it transcribed, it just didn't stop at the virus, it just continued to the next gene, and it added 2 kb of RNA all of a sudden. The 2 kb of RNA went into a virus. The virus then became a retrovirus with an extra piece of DNA, cellular DNA, right? And so that meant that these viruses had the ability to pick up foreign DNA from the chicken, right? And then that gene was isolated from the chicken, right? And because it was the gene that was suspected of causing sarcoma, it actually caused, it actually was called Sarc, right? And because it came from the cell, the normal cell, the chicken normal cell, it was called C-Sarc or cellular Sarc, right? And the one that came from the virus was called V-Sarc because it came from the virus, right? So it was called SRC, Sarc. And there were 2 forms, one that came from the chicken, C-Sarc, and V-Sarc, one that came from the virus, right? And when they were both sequenced, they had 1 base pair change. So the virus one contained a mutation that the chicken didn't have, of course, right? All right. Now, the next real breakthrough came by isolating temperature-sensitive mutants of the virus. So you isolate mutants, what you do is you just keep looking at lots of viruses, and you're looking for viruses that don't grow at 39 degrees and that do grow at 32 degrees, right? They're temperature-sensitive. The way that temperature-sensitive mutation usually works is the thermal aspects of it. So there are amino acid changes or change in the protein, and the protein has to fold. And it has to fold the correct way to work. And at 39 degrees, it's unfolded because of the mutation, and it doesn't work. And at 32 degrees, it's folded because thermally it's easier to fold at the low temperature. There aren't the thermal forces that are breaking it open, right? And so it's called a temperature-sensitive mutant, right? And the temperature-sensitive mutant was in the Sarc gene, and it made the transformation or the formation of the tumor temperature-sensitive. Well, that was a really good line of evidence to suggest that Sarc was causing the tumor, right? So here was a chicken gene that got captured in a virus, had a mutation, and caused the tumor. Very quickly, the function of the protein was gotten to be known. It was a protein kinase, right? Protein kinases are enzymes that phosphorylate other proteins. They're signaling proteins. When they signal by phosphorylating a tyrosine amino acid, they're usually signaling for abnormal growth or very fast growth, right? And that's the phenotype of a malignant tumor, abnormal growth, right? Instantaneous, oh, and I should say one other thing. Sarc is a preserved gene evolutionarily. Right away, people looked in humans for Sarc and found it. Right away, the hypothesis was retroviruses don't cause tumors of humans, but the mutation could, right? Mutations accumulate as a function of age. Mutations accumulate as a function of chemicals. Mutations are inherited, or can be inherited. They don't have to be. They can be spontaneous over a lifetime, or they can be inherited, right? And they make a tumor, right? So this Sarc was the first so-called gene oncogene, right? Or cancer-causing gene. And it was dominant. That is to say, if you went into a chicken cell, it had 2 good copies of Sarc and it had 1 bad copy from the virus. So it was acting in a dominant fashion. So actually, it was sufficient to get 1 mutation in 1 allele spontaneously, and you would have Sarc, right? Now, Sarc was a funny, Sarc was conceptually the first breakthrough in understanding of unifying everything I said and understanding what was happening. So by 1976, everybody and their brother in the field went to go look for oncogenes, right? Just big deal. Let's look for oncogenes. All right. Now, let me say that oncogenes could be found in viruses this way. Oncogenes could be found the way Goff told you yesterday. They integrate next to another gene, and they have an LTR. And the LTR is a promoter. It's an enhancer promoter. So they transcribe the adjacent gene. They don't even have to form a virus. They suddenly are overproducing the adjacent gene. And some genes don't require mutations. If you just overproduce them, which is like a mutation, right? They actually drive a cancer, right? So over the years, maybe 300 oncogenes have been found. All right. So that everyone by the 80s declared victory. We understand cancer. Oncogenes are the cause of cancer. Some viruses cause cancer. 20% of human virus cancers are caused by viruses, right? Bang, right? Answers, done. We thought we were finished, right? However, there were some gnawing contradictions to this, right? Really sort of things that didn't fit, right? And one of them was an experiment, actually, that was done. It was done in 2 places, in Sweden and in England. And everybody got the same result, and everyone could reproduce it. And it was the following. Suppose I took a cancer cell, right? And it's immortal, right? You know, cancer cells are immortal, right? So it grows forever, and I have it in a petri dish in culture, right? And then I took a normal cell, and it's not immortal. It grows for 70 generations and stops. So that's a good definition of having these cells in culture, right? And if I fuse them, I made what's called a heterokaryon. It has 2 nuclei. The nuclei come together. It has double the number of chromosomes. That's a little bit of a problem. But hey, there's the cell, right? You could make the prediction right away. The cancer is caused by oncogenes. The oncogenes are dominant. the hybrid is going to be cancer producing. So if my assay is, do I make cancers in mice, immunologically compromised mice, I should make cancers in mice. And the answer was you don't. The cells are normal. Now that was a sort of annoying observation. The prediction of oncogenes is they're dominant. And yet if you fuse two cells, right? So you say, okay, well, that's a little inconvenient, but maybe I should ignore it, right? Many people ignored it. Maybe it means something, but I don't care, right? So other people said, you know, I want to do some more experiments, right? Suppose I take two normal cells. Doesn't make a tumor. Suppose I take two cancer cells and I put them together. Sometimes it doesn't make a tumor. That was even more annoying, right? So it harkened back to an experiment.
Continues to have a double.
Actually, it loses some chromosomes, and I'll get to that in a minute. But it continues to have the property of either making a tumor or not, right? Yeah. Actually, the first hint of things to come was that it does lose some chromosomes with time, and when it does, sometimes it makes tumors. All right. So just the simplest hypothesis is on one of those chromosomes is a gene that prevents making tumors, right? Okay. So now we go to a guy named Al Knudsen. Al Knudsen was a pediatrician. He was working at MD Anderson in Texas, and he was working with a disease called retinoblastoma, right? Retinoblastoma is interesting. It's a childhood disease. It's inherited, actually. Was not quite known to be inherited except that it was in families, but not that there was a gene isolated yet. Retinoblastoma is a tumor of the retinoblast, which is the cell before the retina cell. And your retinoblasts are born with you, and in about 6 or 7 years, they're all mature and they make a retina, right? And you have no more retinoblasts, so you'll never regenerate your retina again, right? That's why you don't look at ultraviolet light or things like that, because you'll destroy retinal cells and they don't grow back, right? There are a variety of diseases of the retina as well that cause blindness, right? And here's what Knudsen found. Knudsen was really sort of interesting. He found there really are two kinds of retinoblastomas. So I'm going to call the retinoblastoma RB, right? There's a kind where you get it at a very early age. So you're either born with it or by 1 year. So 0 to 1 year of age, right? This, of course, violates the age issue, right? But you get it very early. Remember, you're not going to get it at past 6 or so because you don't have even retinoblasts after 6, right? In this case, when you get it at an early age, it's always bilateral. That means both eyes, right? So the way they cured this was they took the eyes out, right? Now they cure it by lasers, right? And there were multiple tumors per eye. All right, so that was one type. The second type was 5 to 7 years of age, one eye, one tumor. Right? Exactly the same cancer. Exactly the same cancer, yet two very different presentations, right? Anybody want to speculate about the unifying theory here? This is a good class because you guys don't know about this stuff. So anyone who really does know, yes, go ahead. Oh, you don't? No, we'll give it a try.
So what I was going to say is that
Yeah.
in this case, it's suggesting that all of the retinoblasts had something wrong to start with, and they all go wrong at the same time, and this one of them mutates.
Not quite, but yeah. Yeah, I mean, yes. I mean, so here, I'm going to point out on this side, right, it's inherited. Yes, exactly. Right? Inherited is, you're inheriting it as a heterozygote. Means one of your parents has had it, right? And you have one mutant gene and one wild type gene, right? Here, it's sporadic. You have 2 wild-type genes, right? So here, it only takes 1 more mutation to cause the tumor. And you have 10 to the 9th retinoblasts. You're on the edge of 60 mutations every time you divide, right? Information transfer, right? Right. So here, you get it very fast, right? Because mutations occur certainly every generation as the retinoblast goes from 1 cell to 10 to the 9th cells, right? Here, it's bilateral. It's very common, right? Here, it's multiple tumors. It's multiple examples, right? It's not every cell, which was what you suggested, right? No one's homozygote, right? Here it's spontaneous. You have to get 2 independent tumors, right? 2 independent mutations. So it's rare, it's late, right? It's 1 eye and it's a tumor, right? Right. Now, what does that say about this retinoblastoma gene? First of all, let me say the second mutation occurs at a very high rate, about 10 to the minus 3rd frequency. And that's actually so high because of the recombination between the 2. So chromosomes are recombining. That's your somatic recombination, not your germline recombination. However, that happens once, then 10 to the 9th, you got yourself a tumor, right? So what's happening here is really quite interesting. There exists a gene called retinoblastoma which prevents cancer. And when you lose both copies, you get the cancer, right? It's recessive. Does everybody follow that from this? You have to lose both copies. Now, once that hypothesis is on the table, and we can clone genes, and we can isolate kids that have this, right? It's easy. You go to the families, you see that they have 1 mutation that they've inherited, you see that the tumor has both mutations, right? Benign, malignant, right? Both mutations, right? Okay? Or you see that they're wild type, and you see the tumor has both mutations, right? Both alleles are mutant. So you can actually prove the hypothesis by sequencing. And once you've got cloning and sequencing and the families, it's pretty straightforward, right? Suddenly, that makes sense of this experiment, right? Here's a normal cell with the retinoblastoma genes. Here's a cancer cell that doesn't have the retinoblastoma genes because it's a cancer cell, right? And here, the normal actually is dominant over the absence of a gene, right? The absence of the retinoblastoma. Here you lose a few chromosomes, right? If you lose the retinoblastoma chromosome that had wild type on it, you now make a tumor. Is that clear to everybody? Yes? No, it's not actually. Retinoblastoma is in every cell of the body. And it turned out that the first tumor to be made in the body was the retinoblast tumor. So actually, small cell lung cancer actually has retinoblastoma mutations. So lots of cancers have retinoblastoma mutations. You're going to find out that retinoblastoma is a tumor suppressor gene, and tumor suppressor genes are checkpoints on cell division. It is a major stop at G1, late G1, that says go, no go for cell division. And when you pass that stop, you don't even need glucose anymore. You're going to finish cell division, right? And if it's a major stop and it's stopping you, right? Okay. All right. Yes.
I have a question about the semantics of the...
Yeah.
So when you say you lose a gene, in this case...
That means you mutate the gene. You lose the function of the gene. The semantics is, when I say you lose a gene, I mean you lose the function of a gene. It could be a point mutation, in which case the gene is present, or it could be deletion. It doesn't matter. The most important thing is you have lost the function of the gene.
Right, and when you say loss of the function, it means that the transcription or the proteins that are...
You could even make the protein, right, and it wouldn't work. Yeah, it means it doesn't function. It doesn't work. And remember, the way to think of tumor suppressor genes is they are negative regulators of growth, right? If they're negatively regulating growth and you've lost the negative regulator of growth, It's as bad as turning on a positive regulator of growth, which are the oncogenes, right? In other words, there isn't just oncogenes. There's oncogenes and tumor suppressor genes. Yeah. Positive and negative regulators in the cell. Yeah.
I mean, just out of curiosity for it. I mean, by this time, like Jacob and Monod had published together.
Oh yeah, the operon, yeah.
So
yeah.
we knew that negative regulation.
Oh yes. No, negative regulation, if you were brought up in phage genetics and bacterial genetics, you would have found this to be perfectly fine. Actually, most of the people in virology were brought up in phage genetics, and they could swallow this very quickly. Most of the chemical carcinogenesis people and the aging people hadn't the foggiest idea what this meant. That's why I think the virologists sort of won the day, right? Yeah.
So just as a point of clarification,
Yep.
am I right in thinking that the oncogenes are the ones that are tumor growth and tumor suppression is this?
Correct. Yep, you're right. You got it. Okay. Now, there's a third class of genes that are important that I've just sort of bypassed, but you'll see how they work out, right? Throughout your life, in fact, while you're sitting here now, right? Cosmic rays are coming through the room. Nothing is stopping them. They're going through you. And among the things they're doing, where's Sasha? Among the things the cosmic rays are doing is breaking your DNA. Right? Single, double-stranded breaks, a whole bunch of things like that. You've had breakfast this morning. Those of you who had bacon are having a chemical mutagen reaction. So chemistry is coming to bear, right? So we can go down the line. But you are probably getting thousands and thousands, maybe tens of thousands of mutations a day. But don't worry, evolution comes to your comes to you. And it has given you about 12 different DNA repair systems. Each DNA repair system is a series of genes. So there may be 400 genes, 500 genes, that are dedicated to repairing your DNA when they break in various ways. Right? Sometimes the sugar is derivatized, sometimes the base is derivatized, sometimes the phosphodiester bond cleaves. All of those are different ways to repair the DNA, right? And if you have mutations in your DNA repair processes, you're going to leave the DNA damage in the DNA. And when you replicate damaged DNA, you make errors and you get mutations. You get mutations, you get cancer. All right? So people who inherit tumor suppressor genes, nobody inherits oncogenes because they're too dominant, right? They actually abort embryos because they don't develop properly. But people who have tumor suppressor genes inherit mutations that cause cancer. People who have DNA repair defects inherit mutations that cause cancer, and there's spontaneous mutations that cause cancer, right? So there's three classes of genes that cause cancer. Yeah. Three classes of genes that cause cancer, right? DNA repair genes, oncogenes, and tumor suppressor genes. Tumor suppressor genes are lost, right? Functionally lost. Oncogenes are functionally activated, and DNA repair genes are functionally lost. Okay? Yeah.
And you made a quick note to like different sorts of DNA repair systems. Do you see any like overlap between those systems to try and?
Yes, there is overlap. So there's a disease called Fanconi's anemia. The 8 or 9 genes in the signal transduction pathway are called Frank genes, or Frank 1, Frank 2, Frank 3. Frank 4 is BRCA2. BRCA is a DNA repair gene that causes breast cancer and ovarian cancer. That's the more common one you know of, right? Okay, so now we can add all this up. Now we've just put the last 40 years of the 20th century and the first 20 years of the 21st century. We have determined what an oncogene is. Let me just define any of these things for you. I could have a mouse. I could prove it's really an oncogene by doing the following. I could have a mouse and I could add a gene to it, like SOC, and it would cause a tumor, and I would prove that the added gene caused the tumor, right? I could subtract a gene, and I could prove the subtracted gene causes a tumor, right? And I could put a DNA repair defect in, and it would get cancer, right? So I have an animal that I can manipulate genetically, and I can line up all the oncogenes, all the tumor suppressor genes, and all the repair genes. I could take the ones that are most common in human cancers and list the 125 most frequently mutated cancer genes, right? And then I could look at the somatic mutation frequency. Now, this is not inherited. This is all somatic over your lifetime, right? Somatic over your lifetime means it's going to not be your germ cells. You're not passing along these mutations. They're in the tumor, right? So this is the somatic mutation frequency here. And here are the tissue types. You can't read this, and it's not important that you read it. It's just 36 different tissue types, right? This is called a skyscraper plot, right? And now just look at what you see. And what you see is, first of all, some oncogenes and tumor suppressor genes, because that's what this list is. Oncogenes and tumor suppressor genes, DNA repair genes, are very common in cancer. Some are less common, right? That's the first thing to say. Secondly, what's common and what's not common is determined by the tissues. Tissue specificity is not trivial. It's very important, right? That's why a benign mutation doesn't... a gene that causes a benign mutation, that is to say, makes a tumor, but it's not threatening, it's not invasive, it's not going to kill you like a wart, right? Right? That's in the skin. But the same virus in your throat or the same virus in your cervix will cause a malignant tumor. The same gene causes a malignant tumor, right? So you can't by gene classify what's benign and what's malignant. You have to understand the tissue specificity. Okay? All right. So here's a list, and this is just to give you the feeling rather than the facts, right? All right. There are big players that are important. Unfortunately, this is the real sort of sad thing, right? The easy ones to drug are all the ones that are very low, right? All the 1%. So they're class of genes which are 1 to 3%ers, right? They're in about 3% of all cancers. Yeah.
Is that a meaningful fact?
The lack of drugability. I don't know if it's meaningful, but it's sort of sad. I mean, that's why I say it. What the drug companies did, I understand what they did. I'm not even blaming them. What the drug companies did is they drugged the ones they could drug, right? The easy ones to pick off, the protein kinases.
Now we're just, drugability.
Drugability is not the cancer ability. It has to do with the protein, yeah.
But this is basically got to be proteins that have a signaling function.
Yeah, it's actually enzymatic and is common because there's an active site. And it's actually, you know where the active site is. And for example, remember I told you protein kinases, or a lot of oncogenes are protein kinases? They're like falling off a log. There are 500 protein kinases. There are inhibitors for every one of them in the body, right? But protein kinases are 1 to 3%ers, yeah. Right?
My problem is the DNA repair enzyme.
Yeah. Yeah. There are 5 big players, right? That's the other thing, right? So may as well let's get on the board who's a player, right? The single most common gene that's mutated is called P53. It's a tumor suppressor gene, right? And it is a stress responder, right? So what is P53? It responds to stress. What is stress? Almost anything you can think of, right? So all your DNA damage, right, is a stress. Right? You don't want to replicate your cell when it has damaged DNA. So it recognizes damaged DNA, this protein, right? And it shuts the cell down. It's a negative regulator of growth. Shuts the cell down and prevents entry into the cell cycle. And if it gets really bad, if you have very damaged DNA, it kills the cell. Right? So it enforces fidelity by death. Right? That's a great way to just think of the function of this protein. It enforces fidelity by death, right? People who have depression and have high cortisol levels activate P53. So that's psychological stress, right? Even psychological stress is sensed by this. Metabolic stress is sensed by it. Ribosomal damage is sensed by this, right? It will not let you finish cell division if you are under stress, right? That's why it's good to stay cool, right? So that's the biggest, actually that's 50% of all cancers, right? Right? A whole class of proteins called BCL2, BCLXL, right, are negative regulators of apoptosis, right? Apoptosis is the death that p53 brings on, right? And BCL2 stops the death, right? They're bad players. They're oncogenes, right? When they're high, doesn't matter that your p53 is working. It will allow the cell to cause, it will allow the cell to prevent death, right? It prevents the death of the cell, right? There are opposites of BCLXL2, right? Called BID, BAX, etc. Right? So these are the negative regulators. It's a negative regulator of apoptosis. If you think what a negative regulator of cell death is, it's stopping cell death, so it's causing the cancer, right? So it's positive. It's an oncogene. This is a tumor suppressor gene, right? You heard yesterday about MYC. MYC is a big player. Yes?
The P53 is not there and supposed to be. Or is it there?
It's lost.
It's lost.
Lost in the cancer. Yeah. By the way, people, I'll show you, I'll finish up by showing you what happens to people who inherit P53 mutations. It's a tumor suppressor gene, so it's inherited. And it'll have some really interesting lessons for us about inherited tumors. MYC. You heard about this yesterday. This is picked up commonly by retroviruses. Was it one of the earliest oncogenes, right? This is a big, big player. MYC is regulated by retinoblastoma. Retinoblastoma is the negative regulator of entry into S phase. MYC is the positive regulator of entry into S phase. Positive regulator, what? Why? It turns on 1,000 genes that are needed for S phase. Why do you need so many genes? Well, you need all the metabolic products to make nucleotides. You need to make all of the substances that double the mass of the cell, right? And that cause the cell to divide. MYC is the major player, right? And retinoblastoma antagonizes it, right? MYC is a transcription factor, right? To give you the terminology of these kinds of things, right? Another major player is another transcription factor called NF-kappa B, right? So there's just a series of transcription factors that are big players. And finally, probably the second most important gene which are being tested is called RAS. And RAS is in the pathway. RAS is actually a nice protein to think about. All of these proteins are in signal transduction pathways, right? That means that they start on the outside. Let me just take you through cell division for a minute. Right? A mammalian cell, right, when it's going to divide, gets two signals from outside. The first signal is a growth factor, right? That means cell A secretes a growth factor that turns on cell B and says it's time to divide. There's a growth factor receptor, and the receptor signals into the cytoplasm, and after you go through the cytoplasm, you go to the nucleus, you turn on a transcription factor. Okay? So that's a growth factor. But the second thing you need is nutrients. You wouldn't want to start without nutrients. So you have amino acids, nutrient sensors. You have glucose, nutrient sensors. You'd never start a cell cycle with too little glucose, right? If you're a bacteria, like Clyde works with and talked about minimums, right? There's no growth factors in bacteria. They're just sensors of nutrients, right? E. coli is not going to start to divide without glucose. But it doesn't know what a growth factor is, right? So there's a double-edged start. And then it triggers a signal transduction pathway, series of other proteins that relay information, right? And in these, right, are little transistors. In these pathways are protein kinases. They phosphorylate the next protein and turn it on, phosphorylate the next protein. But there also are transistors, which are just on-off switches in the cell. And they're on when they bind a nucleotide called GTP, and they're off when they have GTP go to GDP. So they're just turned on and off by an enzyme that takes the phosphate group off GTP, makes it GDP, right? Diphosphate, triphosphate to diphosphate, right? These are called G proteins, and they're all over the cell, and they're exactly like transistors, just on-off switches. They're exactly like the light switches, right? So these are the components, and that's Ras. Ras is a GTPase, and it has a couple of other proteins that load on the GTP and take off the GDP and so forth, right? Okay, right? Those are the big players. Yeah.
MYC again, a cancerous mutation in MYC causes.
Too much MYC causes you to get into the S phase and just keep going.
So
The mutation is an amplification or an overexpression or a virus that sits next to it that makes too much. There are mutations that turn on the transcription factor, but they're rarer.
So MYC is an onco?
Yes, MYC is an oncogene.
And RAS would be
RAS is an oncogene, serious oncogene. This summer, good news, this summer, the first 3 RAS drugs are going to be tested for cancer. They will be big players if they work. All right, I'm going to finish up. I do want to show you one more thing. These are different tumor types. These are the number of somatic mutations, the somatic mutation frequency per megabase, I think. Yeah, per megabase, right? So one of the things you'll find out, these are called rhabdoid tumors. Rhabdoid tumors are tumors of muscles. They're childhood tumors. Childhood tumors have very low, each point is the number of mutations in the tumor. You get it from sequencing, right? So I'm sequencing all these different kinds of tumors. I'm just counting the number of mutations. Some of them contribute to cancer, some of them don't, right? Mutation is random, selection's not, right? That's what cancer is all about. Mutation is random, selection is not. Okay, so here we have a rhabdoid tumor and a Ewing's tumor. These are childhood cancers. And the number of mutations is very small. Childhood cancers are when you're 3 years old, 5 years old, 7 years old. Doesn't leave a lot of time for mutations. It takes fewer mutations to cause the cancer. All these cancers are malignant. None of them are benign here, right? They'll kill you.
plotting many different instances of
Yes, the plot is every point is a different tumor. Just shows you the variation, right? But it shows you that there's 3 log differences in these tumors, right? The tumors that have the highest levels, right? Melanoma, right? Has the highest level of mutations. Turn out to be the tumors that respond best to immunotherapy, which we'll hear about as we hit immunology, right? And that makes some sense because mutations are foreign proteins in the cell, in the body, and the immune system recognizes foreign proteins, right? As a generalization, if that's the correct interpretation, right? So, yes. Just looking for colon carcinomas, colorectal, yeah. Here, yeah. All right, so colorectal, you see that point that's very high in colorectal? Here's colorectal, by the way. A couple of points that are very high. Those are because they're inherited mismatch repair. So those people have inherited a bad repair system that works in the colorectum, right? And that's contributing, so it lowered the age at which they got it, right? But it raised the mutation frequency, right? So that's an example of DNA repair raising the mutation frequency, right? Okay. If you look at the nature of the mutation, right? Just look at the nature of the mutation. C to T is the most common, yellow. It's the most common mutation. And the reason is that those Cs are usually methylated, and methylated C has a 10-fold higher mutation rate than non-methylated C. And when it's changed from C to T. It's the most common mutation in cancer. I think, Ben, are you going to talk about that at all? Maybe. Yeah.
So you said in these cancers that you might get as a young person, only a few mutations are needed to cause the cancer in young people.
Well,
another way
yeah.
around is like, this is a sort of cancer that you'll get from only a few mutations. So do you see these in adulthood and just throughout life very
No, you don't see them. These are childhood cancers, and you don't see them in adulthood.
What suppresses the few mutations that are needed?
Yeah. So the cells is gone, so what happens? It's like your retinal cells, right? You have cells when you're a child that you'll never have again. I'm sorry to tell you that, even at your young age. Yes. Yes. You'll have cells that you'll never get again, right? And those cells are susceptible. If I'm talking about tissue specificity, those cells are susceptible to some kinds of oncogenes and tumor suppressor genes, and they'll never be in your body again. So your chances of getting that kind of cancer is non-existent after a certain age, right? But this number isn't the operative number of the number of oncogenes and tumor suppressor genes. It's just it's an indication of how many mutations are in the tumor, some of which contribute to the cancer and some of which don't, right? So just take this as an indication that childhood tumors have fewer cancers, fewer mutations that are important. And that's generally true, by the way. I mean, that's correct. Yes. Let's go back here and then here. Yeah.
Is it mutations in the whole genome or just in exons are important?
This is just exomes. Yeah. So this not the whole genomes are a pain in the neck to sequence in this kind of volume. You usually just do exomes. Yeah. Yeah.
So are these big players and those all unique to humans, or can they be studied in any?
Yeah. So the big players will make tumors in mice. We don't know a lot about spontaneous tumors in mice. We don't sit around and watch for them to happen. We actually put these genes back into mice and then they get cancers. They are certainly big players in animals like dogs where you are looking at spontaneous tumors. So yeah, a big player is a big player. Yeah. P53 would be most common in dogs and mice and rats and humans. Can't say more.
Are there any
Unique human cancers? I don't know. Maybe. I don't know. Yeah.
So is there any epigenetical
Yeah.
cause of cancer? So that is
Yes.
due to mutations or
Yes.
mutations on oncogenes
Yes.
and so on that
Absolutely. There are causal genes that are epigenetically regulated genes. So they're changing the transcriptional program in the cell. They're just like transcription factors. Yeah.
so they are not
They are in the list. Yeah.
in the list.
Yes.
They are all
They are either oncogenes or tumor suppressor genes. Yeah.
they are considered to be suppressor.
Yeah. Yeah. Yes.
Is there a reason why only CNT are getting mutated and not A and G?
Yes. The reason that C to T mutations are most common is that the rate of mutation is tenfold higher than all the others. If the rate is tenfold higher, they're the most common. Oh, it's it's based on the chemistry. Sorry, yes. So your when you put a methyl group on C, right? What happens is it makes it sensitive to hydrolysis by water. The methyl group becomes alcohol, and the C becomes a T. No. Physicists don't understand chemistry. That's why I don't even.
Well, I
Right.
can see from the diagram.
Yes.
You know, thymine has a methyl group stuck on it, the cytosine doesn't.
That's right. Yes.
Where does
But
the methylation?
no. But this is methyl cytosine. Yes. Yeah.
The methyl when you methylate a base, you're
Yeah.
methylating not something on the backbone. This thing, while you're actually
No, you're actually methylating the base. Yes. There are 3 enzymes called DNA methyltransferases, and they methylate cytosines.
Do they methylate it on the site that turns it into thymine?
No. No. No.
But it's not big.
Yeah. No, they're both pyrimidines. Yeah. Yeah. All right. Let me just show you. Yes, this is probably. Yeah. Well, yes. This will be useful. So this is a, so now there's some devastating diseases that come about because of these things. Tumor suppressor genes can be inherited, they're recessive. They're always inherited in humans in the heterozygous state, right? So people are born pretty normally, and you have to lose one of the good, you have to lose the good copy, the wild type copy, but that happens at a high frequency, so it acts like a dominant mutation, right? And this is inherited p53 families, right? So they're about, so this is in flux right now in a really interesting way. Inherited p53 families were thought to occur in about 1 in 20,000 people. However, in genetics, there are really great founder effects. By founder effects is if someone who has left a lot of offspring, right, has come in a couple of 100 years ago to some area and left a lot of offspring in that area, and people are still in that area, you may find that the 1 in 20,000 goes to some other number, right? And that happened when a Portuguese businessman came to Brazil in 400 years ago with his 2 sons. And between the 3 of them, they must have fertilized an awful lot of people. Women mostly, yes. They fertilized a lot of women because the number of people with p53 mutations south of Sao Paulo is 1 in 375, right? And these people develop cancers over their lifetime, and they develop up to as many as 20 independent cancers. All right? That's one of the proofs that the absence of this gene, which is a stress responder, this is a fidelity gene, right? This is the best example I know of how important fidelity is, right? It enforces fidelity by death. If you don't have it, you don't have fidelity. You don't have fidelity, you are going to develop oncogenes, tumor suppressor genes, mutations. You're going to make mistakes in the replication cycle, all right? So let's just look at what happens here. First of all, this is the proportion of cancers over a lifetime. Remember I said you can have anywhere between 1 and 20, so it's a distribution over the population. If you look at that distribution on average, most of the cancers, as many as 5% occur very early in life, right? However, they are restricted to certain kinds of cancers, right? This is a choroid plexus papilloma in the brain, right? This is a medullary blastoma in the brain, a rhabdomyosarcoma in the muscle, right? That's like Salk, right? Adrenal cortical carcinoma, right? And just above the kidney, right? Tissue-specific timing, right? The reason that there's tissue-specific timing is these cancer cells have to be stem cells. It is not sufficient. A great example of transforming a non-stem cell, right, or a cell that's able to differentiate is a wart. You get a benign tumor, right? You differentiate the existence of the tumor away. But if you transform the stem cell, it will be a stem cell forever. It can differentiate and make a benign part of the tumor, so the tumor can be half benign and half malignant, but the stem cell always reproduces stem cells, right? And these are the stem cells that exist when you're young. These are the stem cells that exist when you're here, right? This big group here is breast cancer. 50% of the women that have this genetic mutation develop breast cancer, but they develop it between the ages of 20 and 40 as the breast tissue expands and contracts, hormonally regulated, right? Stem cells, stem cells, stem cells, right? Let me just show you the raw numbers. They're great, right? So this is called excess risk. Excess risk is everybody in this room, no one has a p53 mutation, thank goodness. So everyone in this room, right, is normal, and everyone in this room has a certain probability of developing cancer over a lifetime. And I showed you what it was. It's very low until you get older, and then it goes up by 4 logs, right? Anything over that is called excess risk. So we're looking at the ability of inheriting a p53 mutation to contribute to excess risk and giving it a factor, right? And I'm looking as a function of the germ layers. Now, I mentioned this once before. The germ layers are the first 3 layers of tissues that are made in development. Ectoderm will make your skin and your brain, neuroectoderm, right? Endoderm will make your bones. Endoderm will make your gut, right? Your esophagus, your stomach, your colon, your ovaries, your prostate. It'll make, I think I got them all. That's endoderm, right? And mesoderm will make your muscles. It'll make your blood, right? Right? So these are germ layers that are going to make all the tissues of your body, right? The excess risk is totally dependent on the stem cell. 1,000-fold excess risk for carcinomas, right? 2 to 4-fold excess risk for endodermal tumors, right? That's the inherited risk. And the spontaneous risk is exactly the opposite, right? 75 to 100% of ovarian, colon, pancreas, lung, that's all the endoderm tumors. 75% have spontaneous p53 mutations, right? And that's because the order of mutations is an important thing. So the last thing I'm going to tell you is order is important. Stem cells are important. These oncogenes and tumor suppressor genes only work in some stem cells. They don't even work in other stem cells. A great example of a benign tumor is having an oncogene mutation that's not working. If I put a p53 mutation into an endodermal cell, it only has 2-fold risk. It makes a benign tumor. That benign tumor will sit there until 3 other mutations occur, and then it will be the 4th mutation, because 3 others have now occurred. And when it's the 4th mutation, when it's the last mutation, it causes the cancer. Right? So order is important, and the cell type is important. And that determines benign and malignant. Right? Took a whole lecture to get to what your question was. Right? Order and stem cells. Right? And so we're at a spot where we're starting to really understand this pretty deeply, right? But that doesn't help us in curing anybody yet, which really is the next thing. So now, I talked about the origins of cancer. We're going to take a break. A quarter after, I guess, so we have a half hour, right? Rous will talk about progression and evolution of cancers, right? So once a cancer arises, it goes through steps in progression, right? Like metastasis and others. And there's an evolution of the chromosomes, and he works on that area. So let's take a break, and coffee's down here, and I'd be happy to answer questions if I could speak. Yeah, it's okay.